This method enables the user to generate a profile of hepatocyte nuclear DNA content from 2D tissue sections for the study of liver development, regeneration, and chronic disease. This automated high-throughput approach can be applied to all 2D liver tissue sections, providing an internally calibrated readout of nuclear ploidy for all simple circular hepatocyte nuclei.
Changes in hepatocyte nuclear ploidy are associated with aging, chronic liver disease, and cancer. This technique provides a simple means to profile nuclear ploidy in fixed or cryopreserved liver specimens. In situ ploidy profiling methods have considerable potential as research and clinical tools for tracking liver disease progression, as they do not require tissue disaggregation or access to fresh material.
After harvesting the murine liver tissue sample, embed the liver lobule into an optimal cutting temperature medium-filled cryomold and immediately place the mold on dry ice to ensure rapid freezing. To obtain slices of the frozen liver lobule sample, section the sample at a 6 micrometer thickness, placing a labeled polyamide-coated slide over each sample for 5 seconds to let each sample stick to the slide. When all of the slices have been collected, allow the samples to equilibrate for 3 to 5 minutes at room temperature before immunolabeling. At the end of the equilibration,